right, so to recap from the last video, we are in the light dependent reaction kind of stage, and we're talking about the photosystem. So photosystem 2 and then photosystem 1. These both are capturing solar energy in the form of photons, and they're kind of like stimulating, I guess, the pigments, which are going to create the movement of electrons. In the photosystem 2, this is where our reactant water comes in, and that splits. And when that splits, we have hydrogen ions, and therefore that electrolyzes. Remember, hydrogen ions were always going to followed by those electrons. They're going to be there. So the electrons kind of leave that thylakoid membrane. You can see there's lots of other stages that are going on here. Do not expect you to know that, okay? What you do need to know is that the electron transport chain is now taking those electrons and moving them around over to our photosystem 2. We also have ATP that's being created by the ATP synthase for this to happen. And what then happens in photosystem 1 is that, again, more photons are being absorbed from that light energy, stimulating those the chlorophylls. So again, electrons are coming over from the electron transport chain, and this is, they're going to leave again, just like we've seen. And as you can see, there's all these other molecules that are coming into play. Once these electrons leave, once they leave the membrane again, this is kind of the end of the light-dependent reaction. And we're going to move into the next stage, which we call light-independent reaction. And that's going to continue on, with our other stuff here. So I wanted to show you the overall picture. This is kind of what's happening. This is the thylakoid membrane, right? So phospholipid membrane. Here's our photosystem 2. Right, so here's PS2, which we're beginning the process. We can take our water molecule, we're splitting it. What are we getting? We're getting hydrogen, we're getting O2, we're getting electrons. Through the solar energy coming in for photons, the electron you can see is kind of leaving. It moves over to this electron transport chain. Again, there's a lot of things going on here. You're not having to worry about too much. But you can see hydrogen ions are moving into the cell. So again, our concentration gradient is definitely changing consistently when we're talking about our hydrogen ions. That's why the electrons are moving so much. The electrons, as you can see, move across the electron transport chain over to PS1. Same thing's happening. Photons are coming in, going to the chloroplast, etc. It looks like a channel. And then the electrons are moving out. Now, that is the end right here of what we consider the light-dependent reaction. So this is light-dependent. The next portion here is going to move into light-independent, is what we call it. Now, here is that wonderful thing. So I said PS2, PS1, electron transport chain, ATP synthase. This is why that high concentration of hydrogen ions is important. So this is like a turbine, right? And so what happens is we need on the outside here to have ADP plus phosphates. And with that hydrogen ion passing through the channel, this turbine kind of spins. And when it does, it creates movement, and therefore these two molecules smush together and make ATP molecules for energy that this actual needs to happen inside the cell. Now, think of like, if anybody's ever been to Holyoke Gas and Electric near the Connecticut River, where they have the fish ladders. I don't know if I've been to the fish ladders. Basically, they use water turbines, so big, huge turbines underneath that basically are going to spin, right? They're going to spin because of the force of the water coming off the river. And that actually is what creates electricity. There's a lot more to it than that, but that's what's happening. And that's the same idea of the ATP synthase here that creates that ATP from the electrical chemical gradient of all of these hydrogen ions on the inside. So now, since the light reactions are occurring, all right, now we have a few other things that are coming into play. So this is what we call light independent. So we have our electrons, like we saw, we have our hydrogen ions, and then I told you, remember, we'd have that new molecule come in, and that new molecule is NADP. All right, and when we're outside of this thylakoid area here, we add an electron, what we're going to get, so these are our reactants. This is our product. And this is really important because now we're going to move into this next phase. So we're harnessing the sun, okay? We're producing chemical bonds, we're producing ATP, we're producing this NADPH molecule. And all of these molecules are really important because they're considered energy carrying molecules. And now the next phase of this is going to happen in the stroma, so outside of the thylakoid, where we have carbon fixation taking place. So now our carbon's coming into play, our reactants. And we're going to move into what we call the Calvin cycle. And a cycle is just what it is. It moves around in a cycle and produces things and uses things and produces things and uses things, okay? So let's look at what's happening in the Calvin cycle. Now the reactions here are not, these are what we call light independent. That doesn't mean they happen in the dark. That just means that they don't necessarily need the photons right now. We've already used the photons. So again, it's a cyclical series of reactions moving in a cycle. This point in time, we are bringing in our carbon dioxide. So our gas here, carbon dioxide, is our reactant in this scenario. This is what's going to help produce the carbohydrate, the C6H12O6. And this is known as what we call C3 photosynthesis. So there basically are three stages. So we have carbon dioxide fixation, we have reduction, and then we have regeneration of RUBP. So again, this is what it stands for. All you need to know is RUBP, okay? So let's just look at this cycle. So you can see here was all the light dependent stuff, light independent stuff, etc. What happens is we have the NADPHs, but we also have brought our carbon dioxide molecules in. Now, we split that carbon dioxide in the fact that we know in the reaction we have 6, but we basically have 1 set here. Okay, so carbon dioxide will enter in, and there's this wonderful enzyme that you do need to know, which is again going to be part of this process called ribisco. So ribisco jumps on with that carbon, and they hit this first phase that we call carbon fixation. So you'll see ATP is used, okay? ATP is made, it's used. We need 6 molecules for that to work over here. What we get is these kind of 3 phosphoglycerate molecules that are going to continue on. Here are more of our reactants, okay? So here we have the 6 NADPHs, which we made from the light dependent reaction. It's going to split those into reactants of NADP and then our hydrogen, okay? And then this is the second phase, phase 2, reduction. So we're going to take this big molecule and we're going to separate it. And this is where we get G3P. So we have G3P, just one molecule of that, okay? So that part of that leaves, okay? So what I should say, there's not one, there's actually 6, okay? And one of the 6 leaves. The rest of the 5 are going to continue on. But we've made that. So again, we had 6 here, 6 here, 6 here. Because again, remember, we put in a certain amount of carbon dioxide. We had 3 molecules of it at one time. So one of these leaves. And so one of these G3Ps leave, and that is creating half of our glucose. So half of our glucose molecule. But that just means that we have to do something else. So this is going to continue. These 5 are going to continue. So there's 5,3-GP are going to now go to this regeneration with our UBP. You can see we need some more ATP coming in. And they're basically going to continue. So our UBP has now continued, and they're going to come back up to this cycle. Now the problem is we only have half of the glucose. So what needs to happen again? Well, we only used half of our carbon dioxide. So it means that this needs to go around this cycle twice. So to use up all the carbon dioxide and create a whole entire glucose molecule, we need to basically go around this cycle twice for every time we have that one kind of chemical molecular equation. When it goes back around the second time, again, 5 are going to continue on and 1's going to leave. And when it does, there's the other half of our sugar. So like I said before, we have 2, okay, 2 of our 3 carbon G3P molecules, and those guys are going to turn into the glucose, okay? So this is, again, very, very vague. It's just kind of walking you through what's happening. So why is it important? Well, it's important for that G3P molecule, okay? That is the molecule that's converted into many other things. It's a hydrocarbon skeleton, so it can form a lot of things: fatty acids, glycerols, simple sugars, glucose, or specifically fructose, starch, amino acids. So it's really important. In our scenario, obviously, we are looking at making the glucose phosphate so that we can make glucose. The crash course video that you are going to watch will also go into this much more in depth. It will basically break things down a little bit, but you got to remember this is a very, very complex process. So between looking at this video, reading your lecture text, and then also listening to the crash course video, you'll be able to see what's going on. So this is what we call the carbon cycle. And that's really important. Carbon cycle is really important. So it kind of gives you this whole idea of here's CO2 in the atmosphere, here's other emission things, and we need that for the plants. Sun need that for the plants. Decaying organisms need that. Fossils need that. So it's just this cycle that we call the carbon cycle, which is really important for all of us to live. And so again, that's why sometimes you can have islands of trees in the middle of nowhere, but they have the water and the sunlight they need to thrive on an island like that. So this week, the last thing that you're going to do this week is a graphic organizer to kind of organize those different stages and what reactants are going in, what products are coming out, and where is it taking place. Now, I want to tell you something. If you're looking at the first line of that photosystem 2, whatever reactants go in, you have products. In the next stage, usually the products are now the reactants to continue this process. So I just want you to be aware of that and do that last, because that's going to take some time for you to really kind of work through and figure these things out. What are the reactants? What are the products? Where is it taking place? All right, folks.